Hello all, let's look at question number 9 from GATE Biotechnology 2020 paper. So the question is, during a positive negative selection process, transformed animal cells expressing what are killed in the presence of ganciclovir in the medium. So option A, pyruvate kinase, option B, viral thymidine kinase, option C, viral serine or threonine kinase, option D, viral tyrosine kinase. Before answering the question, let's learn about the technique that uses this kind of a selection method. So, gene therapy is an experimental technique that uses genes to treat or prevent a disease. So, to treat a disorder by inserting a gene into a patient's cells instead of using drugs or surgery is the basic principle. So, the first attempt at modifying human DNA was done in 1980 by Martin Klein, but the first successful nuclear gene transfer in humans was approved by the National Institute of Health in 1989. There are two types of gene therapy, somatic cell gene therapy and germline gene therapy. So, in somatic cell gene therapy, the therapeutic genes are transferred into any cell other than a gamete, germ cell, gametocyte, or undifferentiated stem cells. So, any such modifications affect the individual patient only and are not inherited by offspring. So, somatic gene therapy represents mainstream basic and clinical research in which therapeutic DNA is used to treat the disease. So, in like simpler words, the change that is made in these somatic cells is not passed on to the offsprings, but it's only reflected in the individual to whom you do somatic gene therapy. And then in germline gene therapy, the germ cells, that is the sperm or ovum, are modified by the introduction of functional genes which are integrated into their genomes. So, this would allow the therapy to be heritable and passed on to later generations. So, even in somatic cell gene therapy, there are different types like ex vivo gene therapy, in vivo gene therapy, and antisense gene therapy. So, in ex vivo gene therapy, you first collect cells from an affected individual, then you correct the genetic defect by gene transfer, and then select and grow the genetically corrected cells, and then either infuse or transplant them back into the patient. So, all these modifications are done in the patient's cell but outside the patient's body. And here's a visual representation of ex vivo gene therapy. The cells are removed from the patient. So, meanwhile, in the lab, a virus is altered so that it cannot multiply or reproduce. And then a gene is inserted into the virus, and the altered virus is mixed with cells from the patient. So this leads to the modification of the patient's gene by the viral vector. So this and then so all the cells express the modified gene. So these modified cells from the patient become genetically altered and they are infused back into the patient. Let's now look at a detailed procedure of generation of a knockout mouse where a disease causing gene is knocked out or removed, like made ineffective, by targeting them, which is similar to gene therapy in humans. So, gene targeting by homologous recombination in embryonic stem cells is a multiple step process. So, the first step is to generate a targeting vector, which is then transferred by electroporation into the ES cells. The ES cells are cultured and analyzed for the presence of the homologously recombined DNA sequence. The target ES cells are then injected into the blastocyst stage embryos. Now, what are these ES cells? So, these are nothing but embryonic stem cells. So, embryonic stem cell, these are undifferentiated cells isolated from the inner mass of a blastocyst. So, the crucially important factor about the progenitor cells of these early embryos is that they are pluripotent, that is, which means they have the potential to differentiate into any cell type, including the germ cells of the embryo. These embryonic stem cells in culture remain undifferentiated as long as they are grown well separated from each other. So it's been found that the presence of the cytokine leukemia inhibitory factor is essential to ensure that these embryonic stem cells do not differentiate in the lab. So, for this reason, embryonic stem cells are generally grown on a feeder layer of fibroblast cells which secrete this LIF into the culture medium. So, most ES cell lines currently in use have been derived from 129 strain of mouse, which has an agouti coat color genotype that is useful when identifying chimeric mice. So this is what embryonic stem cell colonies growing on a layer of fibroblast feeder cells looks like. So these are healthy, undifferentiated embryonic stem cells. They are well separated from each other, so they don't differentiate into other cell types. Let us now look at the steps involved in generating this knockout mouse. So the first step is to generate a targeting vector. So, when designing and constructing a target vector, a number of factors must be considered, which will influence the type of mutation to be introduced, the efficiency of targeting, and the ease at which successful targeting can be detected. So, the first thing is that the vector has to deliver the mutation and it has to target the correct part of the genome, and it has to cause the mutation in the right part of the genome, and it has to be easy, and it has to be like it should be able to easily identify the targeted cells, the cells like which have undergone the mutation. uh, yeah, so these are the main characteristics of a targeting vector. So for successful and efficient targeting, the vector must contain at least 5 to 10 kb of isogenic DNA homologous with the sequence to be targeted. This homologous sequence is divided between the short arm and the long arm of homology, and this permits easy screening of the ES clones. So, it is ideal to identify gene targeted colonies by PCR design to span the short arm of homology. So, it is known that the efficiency of homologous recombination is decreased when there are base pair differences between the donor and recipient DNA. So for this reason, it's a common practice for the DNA used to construct the targeting vector to originate from the same mouse strain as the embryonic stem cells. And then there are positive and negative selection cassettes. So this selection means that you just choose the cells that have undergone the mutation or whatever you design to target, like whatever change you expect. So you have to just screen for the positive cells and you have to select them. So since gene targeting by homologous recombination occurs at low frequencies and the targeting construct is much more likely to randomly insert anywhere into the genome, it is essential to be able to screen the successful ES colonies quickly and efficiently, like for the successful targeting. So for this reason, most targeting vectors will be designed to insert a positive selection cassette into the gene of interest. And apart from positive selection, negative selection also allows for the enrichment of cells that have undergone the desired genetic modification while eliminating the cells that have not been modified. So this selection process ensures a higher likelihood of obtaining cells with the intended gene knockout. The most common positive selection marker is the neomycin phosphotransferase, the NEO-R gene, which, when expressed in the ES cell genome, will render the cells resistant to treatment with the antibiotic rheomycin sulfate. And a negative selection marker, the HSV thymidine kinase, HSV-TK gene, can also be used to enrich for gene targeted colonies. The negative selection marker is cloned outside the homologous sequence in the targeting vector and will therefore not insert into the genome when the homologous recombination occurs. For example, the herpes simplex virus thymidine kinase gene, the HSV-TK gene, when expressed in ES cells, will produce a toxic product in the presence of ganciclovir, which is a drug, and so it kills the ES cells that express the HSV-TK gene. So again, ganciclovir is an antiviral drug. It is used to treat infections caused by viruses. So ganciclovir is used to treat the symptoms of cytomegalovirus infection in the eyes in people whose immune system is not fully functional. So this includes patients with acquired immunodeficiency syndrome, which is AIDS. And like to summarize negative selection in simple terms, let's say in a gene knockout experiment, we introduce like a gene conferring resistance to a specific drug or an antibiotic is introduced along with a gene knockout construct. So, cells containing the introduced gene knockout construct are subjected to selection pressure. So, the pressure usually involves the application of a drug or antibiotic for which the introduced selection marker provides resistance. So, the drug or antibiotic is toxic to the cells that did not undergo successful, like the gene knock in. So, the cells where the antibiotic resistant gene has not been experimentally introduced, they are not resistant to the drug. So, on application of the drug, these cells die. So, therefore, the cells that still have the intact target gene or have not incorporated the knockout construct will not survive in the selection process. So, here's a schematic of a targeting vector. There are two homology arms which flank a positive drug selection marker NEOR and a negative selection marker HSVTK, and this is placed adjacent to one of the targeting arms. So, a unique restriction enzyme is located between the vector backbone and the homology arm. Let's now also look at the rest of the process after the positive negative selection. So, the next step is ES cell transfection. So, the most efficient method for introducing the targeting vector into the embryonic stem cells is by electroporation. So, the linearized vector DNA is electroporated into a large number of ES cells in a single cell suspension. The cells are then plated onto fresh feeder cells. So, then 24 hours after electroporation, the selection process can begin, which will kill cells which have not incorporated the targeting vector by homologous recombination. So, the embryonic stem cells are cultured in the media containing the drugs used for selection for 7 to 10 days. So, this will enrich the population with cells that have undergone homologous recombination. So, however, it must be noted that the process is not 100% efficient. And then let's look at gene targeting by homologous recombination. So, homologous recombination is a DNA repair mechanism that is employed in gene targeting to insert a designed mutation into the homologous genetic locus. So, the targeted homologous recombination can be performed in murine, that is mice embryonic stem cells, through electroporation of a targeting construct. The technique of gene targeting by homologous recombination allows for the introduction of engineered genetic mutations into a mouse at a determined locus. The most common application of gene targeting is to produce knockout mice where a drug resistance marker replaces an essential coding gene in the genome. So again, like just here's a representation. So here's your target gene, and here are the selection markers, and these are the recombination sites. And then after the procedure, homologous recombination occurs and the gene is knocked out, and yeah, so you have the selection marker. So the mutated locus is resistant to these drugs G418 and ganciclovir. So homologous recombination results in the transfer of only the neomycin resistance gene to the host cell. The next step is to identify the embryonic stem cells targeted by homologous recombination. So, to identify these ES cells that have undergone gene targeting by recombination, discrete colonies are identified and picked. So, the colonies are dissociated into single cells by treatment with trypsin, divided between two wells on duplicate microtiter plates, and are cultured. The purpose of dividing the cells between duplicate plates is to allow one plate of cells to be used to prepare DNA to identify the targeted ES cells, and the other cells from the second plate can be used to inject into blastocysts. So, genomic DNA is prepared from each ES cell clone, which is then screened by PCR to identify clones in which the recombination has happened. So, positive clones must then be further analyzed, usually by Southern blotting and DNA sequencing, to verify that all regions of the targeting vector have undergone the desired recombination event. And then the embryonic stem cells are injected into the blastocysts of the mouse. So blastocysts, which are 3.5-day old embryos, are collected from the uterus of donor female. It is usual when the embryonic stem cells from the 129 strain of mouse to collect blastocysts from a C57B16 mother. So this mouse line has a black fur coat. So ESLs carrying the desired mutation are treated to give a single cell suspension. The ESLs are drawn up into the injection pipette by gentle suction, and the blastocyst to be injected is held by suction on the holding pipette. So the injection pipette is advanced into the cavity of the blastocyst, which is known as the blastocele, and 10 to 15 ESLs are released. And after injection, the embryos are cultured for a few hours to allow them to re-expand slowly before being transferred into the uterus of the pseudo-pregnant foster mother. So this is how the modified embryonic stem cells are injected into the blastocyst. So the blastocyst is held on the holding pipette by gentle suction. So this is the holding pipette. It has a slight pressure that sucks, suction pressure, so that holds your blastocyst in place. So the injection needle containing the embryonic stem cells is advanced into the blastocyst cavity. So it goes into the cavity, and this is the blastocele. So here the embryonic stem cells are released. So the injection needle, after the cells have gone inside the blastocele, the needle is then slowly removed. The last step is to identify the chimeric mice and breeding to generate the homologous mutant, that is a knockout mice. So approximately 1 week after the mouse pups are born, the coat color becomes apparent. At this stage, it is possible to identify agouti from non-agouti coat color. So it is therefore possible to identify chimeric mice by the coat color if ES cells from the 129 mouse strain have contributed to the development of C57B1 embryo, which is the non-agouti. So these are black mice. So which mice have this color and which ones are the knockout mice. Embryos in which the ES cells had made no contribution would appear as the wild type black mice, whereas those in which the 129 embryonic stem cells had made a contribution would contain a certain level of the agouti color coat. So the chimeric mice therefore contain some cells carrying the targeted mutation on one allele and other cells which are wild type. So to generate a gene knockout mouse, it is essential that some of the germ cells carry the targeted mutation. To test for germline transmission of the mutation, chimeric mice are bred into wild type mice, and if the germline transmission occurs, a proportion of pups will be heterozygous for the targeted mutation. So heterozygous mice can then be bred to produce mice homozygous for the targeted mutation, which is the gene knockout mouse. So again, here's an overview of the whole selection process. You first electroporate the targeting vector into the embryonic stem cells that have positive and negative selection markers, and you then inject these the selected embryonic stem cells into the blastocyst, and then the embryos are transplanted into the foster mother, and then once the mice are born, you identify the chimeric pups by the coat color, and then breed the chimera with wild type mouse and test for germline transmission of targeted mutation. Now coming back to the question, during a positive negative selection process, the transformed animal cells expressing what gene are killed in the presence of ganciclovir in the medium. So we just saw that ganciclovir is an antiviral drug, and the cells that are not resistant to this drug will die. So basically, if they express viral thymidine kinase gene, they release some toxic substances in the presence of ganciclovir, and so they are killed. So the answer to the question is viral thymidine kinase.